Okay, I'm assuming you can hear me. I'm going to give this a try then. I'm sorry I can't be there in Armenia. I've been looking forward for this for 2 years, but we're living in a strange world. I was going to give you a talk here on some of the broad work we're doing on what we call biology scaling laws. So most of us are attracted to biology because of diversity. This is just an example down below of a bunch of different ciliate species. They also range tremendously in size. And one of the things we're interested in biology is trying to develop general explanatory hypotheses for variation in traits that are independent of the details of the species, but scale with size. And two famous ones are illustrated here that some of you may be familiar with. On the left is a scaling. These are log plots. And on the left is a plot of the average population rate of increase as a function of the size of the organism. There's about 20 logs here. So this is a very general relationship. To the right is the metabolic rate per unit weight in animals. And often these plots will have straight lines on a log-log scale. And sometimes they'll have exponents like this one here and this one here, close to minus 0.25. And people have spent a lot of time trying to understand these relationships and argue that they're based on basic biophysical principles. Often the scaling relationships aren't exactly 0.25. The one on the right, you can see, actually has an accelerated rate here. So often these arguments are not entirely general. There's been a lot of debate about them. But all of the prior work in this area and many others have assumed that natural selection drives these kinds of patterns. And although natural selection is the most powerful force in the biological world, it's not all powerful. And what selection can and can't do is dictated by what we call the population genetic environment. So mutation creates the variation upon which natural selection operates. Recombination reassorts variation within and among chromosomes. Random genetic drift is sort of the lens on the evolutionary process. It's the noise in the evolutionary process due to finite numbers of individuals and our chromosomal architecture. So the unifying theme underlying everything I want to talk about today is what I call the drift barrier hypothesis. And the math can get quite complicated, but I think the verbal explanation here is quite simple. So imagine we have some trait that's under relentless positive selection to increase. And it's striving to get up to this dashed line. That's the best trait performance you could get based on biophysical principles. And what we're looking at is what would happen in populations of different size. So population size is very, very large. Natural selection is quite efficient, and you might get close to the biophysical limit. But there's still some drift operating to prevent selection from being fully efficient. And then as you move to the left, population sizes are much smaller. There's much more noise in the evolutionary process. And so you end up having this gradient of mean trait performance, whatever the trait is, mutation rate, metabolic rate, whatever, as a function of population size. You'll get these gradients, even though the strength of selection and the basic ecological features are the same in all these cases. So what you might see, for example, if you had the luxury of studying populations over time, you had a population of, say, 1 million individuals here. We're looking and we're censusing it every 1,000 years or something like that. And there'd be this long-term distribution of mean phenotypes here, and that the long-term average would be here. And then as you go to larger and larger population sizes, these distributions shift to the right. So you're getting this gradient of increasing trait performance with increasing population size, which, as we'll see, happens as you get decreasing organism size. So to make clear this phenomenon of random genetic drift, I prepared just a simple cartoon here to explain how noise arises in the evolutionary process. So here we have a small population of, say, 10 individuals. This is a gene locus with two types of alleles, the black one and the white ball here. And then the population produces a bunch of gametes, and now we resample for 10 new individuals in the next generation. And the frequency has moved from 0.5 to 0.6. And then repeat this another generation. Now it's jumped to 0.8, and then another generation. Now it's gone down to 0.4. Eventually, either the black allele or the white allele will be completely lost due to chance, and we call that fixation in the population. And what's important to realize here is that this principle applies to populations of all sizes. It's just that the fluctuations in allele frequencies change with the population size. So as an example, we start again with a population with an allele frequency of 0.5. The population has 25 individuals, and you see that the allele frequency wanders around and eventually it hits a value of 1, so it's gone to fixation. Now we have a population that's 10 times larger, 250 individuals. And again, you get this wandering around, but you see that the fluctuations are smaller in size. And here we have a population of 2,500. Again, there's these fluctuations, but they're still smaller in size. But even in these large populations, we'll eventually get to the point where we get complete fixation or complete loss of an allele. And the time scale that's important here is directly proportional to the population size. So larger populations maintain more variation over longer periods of time, which I'll get to in a second. The numbers of individuals aren't really the main thing that drive chance in evolution. Probably the main thing is the fact that our genes are all glued together on chromosomes, which causes interference between allelic variation at different genes linked on chromosomes. So here we have an example of, I guess, 10 chromosomal segments in a population. Each tick mark is a variant locus in the population. And what's happened here is a beneficial mutation has arisen here, this blue ball, and it's so beneficial that it pulls this chromosomal segment through the population to fixation. And when it does this, all the other variation in the population is lost. This is called a selective sweep. So this is good. A beneficial mutation is fixed, but a couple of other things that have happened here. So this tick mark that was here, these red balls here, these were actually, this was a beneficial mutation as well. It was not as good as this beneficial mutation. So it's outcompeted by the blue ball here. On the other hand, this tick mark here, the green ball here, that's a deleterious mutation. It was dragged to fixation along with this beneficial because it's not doing as much harm as this one is doing good. So in the end, due to linkage of these genes, we have a less efficient response to selection. We fixed one beneficial allele, we fixed one deleterious allele. But if there were free recombination between all of these, we would have left the deleterious allele behind, and we would have fixed both beneficial alleles. So in this sense, recombination inhibits the rate of progressive evolution. This becomes important because recombination rates vary dramatically among different phylogenetic lineages of organisms. In fact, there's an inverse relationship. This is the first of these scaling relationships I'll be showing you. There's an inverse relationship between the recombination rate per base and the genome size. And there's a simple reason, physical reason for this. In all organisms that we know of, there's typically no more than 1 or 2 crossovers per chromosome arm. And as you grow larger and larger genomes, it's not usually because you're adding more chromosomes, you're just increasing chromosome size. So if you double the chromosome size, you reduce the amount of recombination per site by 50%. There is some variation in chromosome number, and that explains the range of variation here. But if you know genome size and if you know the number of chromosomes, you can predict about 99% accuracy the average amount of recombination. So the bad news is us things down here, vertebrates and land plants, very, very large genomes. We have very low recombination rates as well as small numbers of individuals. And this means that natural selection is particularly inefficient in these phylogenetic lineages compared to things up here, for example, yeast and other unicellular eukaryotes, where there's a lot of recombination, making selection more efficient. Now, so there's a lot of stuff going on here in terms of our trying to understand effective population sizes. And so how can we do this? We can't really go out and count individuals in most species. And so we do this in an indirect way. We try and infer from standing levels of genetic variation the size of the population that must have been responsible for maintaining that variation along with the mutation rate. And we do this typically by looking at third positions in codons, codons for amino acids that are what we call fourfold redundant. So for example, for threonine, one of the amino acids, these are the 4 codons. And it doesn't matter if you have an A, C, G, or T in the third position, you still get threonine. So we like to think of these as neutral sites. And if they're neutral, that means that the only thing governing the variation is going to be mutation, bringing in new variation, and drift, eliminating variation. And so if we think about the standing level of variation, you think about 2 sites that are identical on 2 chromosomes. There's 2 chromosomes, each mutating at rate U per nucleotide site. That's the rate of input of variation. 1 over 2 NE, this is the power of random genetic drift. NE is the effective population size. That's the rate of loss. If you keep the population size constant and the mutation rate constant, this will reach an equilibrium level of genetic variation, just the ratio of the input to the output, and that's equal to 4 NEU. This is really useful because we can measure the amount of variation at silent sites in natural populations by sequencing large numbers of individuals and just observing the nucleotide heterozygosity. And if we know, we then set that variation equal to this quantity here. If we know the mutation rate, which we do, as we'll see in a few minutes, you can factor out 4U and you can get an estimate of the effective population size. And we've done this for quite a large number of species now. And this is the sort of take-home message in terms of how effective sizes of species vary with the average size of the individual. It's almost 20 orders of magnitude differences in size of organisms. As you can see, there's an inverse relationship. So things that are very, very small, bacteria, have very large NE, and things that are very large, like us vertebrates, have relatively small NE. This is important in terms of natural selection for the following reason. So suppose you're down here, you're a species with an effective population size of just 10,000 individuals, which is historically the effective population size of humans. The way to think about this is to take the reciprocal of this. That's the power of random genetic drift. And that tells us that if you're way down here, any deleterious mutation with an effect smaller than 10 to minus 4 can't be seen by natural selection. Likewise, any beneficial mutation with an advantage less than this is also invisible to selection. So there's a lot of mutations we can't take advantage of. And then we move up here to the top, bacteria. Let's say the effective size is 10 to the 8th. That means bacteria can take advantage of beneficial mutations with effects as small as 10 to the minus 8. So 4 orders of magnitude range of variation that microbes can use in responding to selection that we cannot. So evolution is much more granular in things like land plants and metazoans than it is for bacterial populations. But no, even bacteria, the effective population size never gets bigger than 1 billion individuals. And I think you all know that it doesn't take more than a few drops in a culture in the laboratory to make a billion individuals of a bacterium. And this is telling us again that there's a big difference between the census size, the counts of individuals, and how the population behaves genetically. And these arguments become important because, in fact, many, many mutations have very, very tiny effects. There's various ways to get at the distribution of mutational effects. This just happens to be from some of our work on Daphnia and population genomics, where we sequenced a few thousand individuals now. And I won't go into the details, but you can end up getting a a measure of the distribution of deleterious mutation effects relative to the effective size. And again, the benchmark here is when this quantity equals to 1. Any mutation to the left of here is so small that it's overwhelmed by drift and natural selection can't utilize it. And what you can see here is a large fraction of mutations in natural populations are in this effectively neutral range. They're not neutral in an absolute sense, but natural selection can't perceive them. We can also get this question from first principles without worrying about population genetics. So suppose you were thinking about a single base insertion into a genome. Could an organism actually feel that? There's no function for this additional base insertion. Could natural selection actually eliminate it? What would it cost? Well, we know from biochemistry to make a base pair, it costs about 100 ATPs. And I'll return to this later, but we know, for example, for an E. coli, to build an entire E. coli cell, it costs 10 to the 10th ATPs. So the ratio here is 10 to the minus 8. So relative to the entire energy budget of a bacterial cell, one base pair addition has a selective disadvantage of 10 to the minus 8, which is right on the edge of what I was showing you would be perceived by natural selection. So bacteria can feel even insertion of just one base in the genome, and in some cases eradicate it by natural selection. So now I've given you a primer on population genetics, which is essential to understanding some of these other scaling relationships I want to talk about. So what I'm going to show you now is that the population genetic environment, in particular, the reduction in population size that accompanies increased organism size, it leads to a dramatic increase in the mutation rate across the tree of life. It leads to a passive expansion of genome size, gene number, and gene complexity. It leads to a reduced maximum growth rate of organisms. And then if I have time at the end, I'll show you one thing it doesn't explain is variation in protein structure across the tree of life. So let's start with error rates inside cells. I'll talk mostly about error rates at the DNA level, replication errors. And what we're going to see is that these rates vary by about 10,000-fold across the tree of life among different species. And I'll talk a little bit about errors that occur inside transcripts, inside messenger RNAs, and we'll see that these are inflated by 1,000 to 100,000-fold relative to our more familiar DNA level error rates. To understand what's going to be going on with the data, we need to think about how selection operates on the mutation rate. So imagine we have a population of cells and everybody's got one deleterious mutation or 0. The deleterious mutations are illustrated by the red dots here. And now a mutator strain arises. It's got an elevated mutation rate, it's reproducing clonally, and over time it will build up an elevated load of deleterious mutations compared to these other non-mutators in the population. Delta U is the increased rate of deleterious mutation arising in these individuals. Each one has a selective disadvantage of S. So this is, again, one of these input-output sort of formulae. And if we let things go long enough, this mutator strain will build up an elevated mutation load, which is equal to the ratio of the input to the output. So that's the excess number of deleterious mutations linked to the mutator allele. But we need to know how much pain the mutator line is feeling, so we need to multiply by the effect of each of those mutations, S. We have an S in the denominator, S in the numerator, and the S, the product, cancels out here. And this tells us that the selective disadvantage of a mutator strain is simply equal to the increased rate of deleterious mutation across the genome. It's independent of the effects of the individual mutations, because if the effect is larger, mutations won't reach as high a frequency. So this is likely to be quite a small number because there are hundreds, if not thousands, of genes influencing the mutation rate. And if we're just talking about a change of one amino acid, this could be quite a small effect. And that's why the drift barrier becomes important. So the way to think about it would be, suppose we have a population of effective size of 100 100,000 individuals. And let's do a thought experiment here where we start out with a population with a really high mutation rate. And so you can think about different mutation rate classes in the population. And going from here to here, these are mutator alleles, and going from here to here, these are anti-mutator alleles. And these are constantly arising in natural populations. So this is bad, a starting point here, but gradually anti-mutator alleles arise and you get down into this gray area and we stay there forevermore. Here we start in the gray area and we stay there forever. Now here we start with a very low mutation rate, which is good, but natural selection is not capable of holding on to it. And it gradually drifts back up into this gray area, into this drift barrier. Now we multiply the population size by a factor of 100. Natural selection is more efficient. The numbers on this axis are smaller. So natural selection is driving the mutation rate down to a lower rate, takes a longer period of time because the mutation rate itself is lower. But the point here is the prediction is there'll be an inverse relationship between the evolved mutation rate in a species and the effective population size. So how to get mutation rates? In the old days, it was done by things like reporter constructs and so on, which are very, very inaccurate. Today, we're in the world of whole genome sequencing. So the way we measure mutation rates are with what we call a mutation accumulation experiment. They're quite conceptually simple. You start with a single individual here. This is a paramecium. She reproduces clonally, and we set up 50 different lines. And then we take each line through a bottleneck of a single individual, generation after generation after generation. We typically will go for 3,000 or so bottleneck generations. They all started out genetically identical. Over time, each line is picking up new mutations independently. At the end of the experiment, we sequence the genomes of all of these lines and then compare them for the newly arising mutations to get the mutation rate and the whole molecular spectrum of mutations. We've done this with quite a few different organisms, ranging from our favorite Daphnia to paramecium. We've done yeast, slime mold, Cenourbatus elegans, Arabidopsis, and so on. We've done now about 40 different species of bacteria of all different genome sizes as well. And I'll just try to give you the basic picture here at the end of one of the experiments. So Bacillus subtilis, for example, 50 lines we did, there's 50 concentric circles, each dot's a mutation. The experiment went for 5,000 generations. We found 450 mutations. We know the number of sites, so we can get the mutation rate per nucleotide site per cell division. It's about 3 times 10 to the minus 10. So this is a really tiny number. This is why we have to do so many lines and sequence across the whole genome. To the right is the smallest genome we can grow on a plate in the laboratory. This comes from a bacterium called Mesoplasma florum. Its genome's less than 1 million bases. We had to stop this experiment early because it was clear from our streaking and plating and bottlenecking, the cells were getting sick. We stopped after 2,000 generations. We found 600 mutations. So this bug here has a mutation rate of about 10 to the minus 8, almost 100 times higher than Bacillus subtilis. To give you an idea how high that is, that's equal to the human mutation rate per human generation, even though it has a generation time of only about 30 minutes or so. This was the 30th anniversary of a famous paper by Jan Drake, published in 1991. At that time, he summarized everything we knew about mutation rates. And there it all is at 6 data points. And this was not done with the kind of experiments I just mentioned. This was done by using reporter constructs, which are quite inaccurate and so on. But the point he made was that it looked like there's an inverse relationship between the mutation rate per base and the size of the genome. This is a log scale here. So every increment is a 10-fold increase in genome size. The slope's -1, so the product of genome size and base mutation rate per site is a constant. That constant is 0.003, became known as Drapes Rule. And that by that 0.003, what we mean is about 3 in 1,000 baby microbes would have a single new mutation per generation. And Jan didn't know why this was occurring, but he felt that this was a very general relationship to be expected, determined by what he called deep general forces. Today, we believe these deep general forces are nothing but the laws of population genetics. And we can go now to our modern data with whole genome sequencing and see if this relationship still holds up. Here's a bunch of unicellular eukaryotes, bacteria. The slope is still -1 with modern data. Looks really good. It would make Jan really happy if you're still with us, but it doesn't look so good when you add in data for multicellular eukaryotes, including us humans and chimpanzee and Daphne and fruit flies and so on. This isn't even running in the same direction. So what's going on here? Well, the drift barrier hypothesis is not predicting any association with genome size. What we're predicting is there should be a unifying inverse relationship with the effective population size of the species. So now I'm replotting the data and changing the x-axis from genome size to effective population size. And now you can see that everything is lined up in the same fashion. There's an inverse relationship between mutation rate per base and the effective population size. You can also see that unicellular eukaryotes have much lower mutation rates than bacteria do per nucleotide site. And there's a reason for that, and that is that eukaryotes have many more genes, much more functional DNA than do bacteria. And remember, natural selection's operating on the mutation rate per across the whole genome, not on a per nucleotide site. So if we now change the y-axis by multiplying the number of functional sites, then the y-axis is the total deleterious mutation rate per genome. And now everything lines up quite nicely. These dashed lines are slopes of -1. There's probably some bias down here as indicated by these arrows, but all these data are quite consistent with the drift barrier hypothesis. So this is really our first general evolutionary genetic explanation for the distribution of any trait at all across the entire tree of life. Now, there are many other errors that can occur inside cells. This is the error at the DNA level. These are heritable errors. But another error that's turns out to be quite important is the rate at which we make errors when we're making messenger RNAs. When we're copying our DNA into RNA, you can get ribonucleotide misincorporations. I won't go through the methods on how we estimate the error rates in transcripts. I'm just going to give you the final line over here. Is for these different organisms, mouse, fruit fly, worm, Arabidopsis, yeast, some bacteria, and so on. This is the ratio of the transcription error rate per site relative to the replication rate at the DNA level. And you can see these range from about 1,000 to 100,000 fold. So there are enormous rates of errors being introduced just spontaneously in our transcripts every day, such that about on the order of 1 to 5% of all transcripts in our cells have at least one error. This is also consistent with the drift barrier hypothesis because transcripts, unlike DNA, are not heritable across generations. Most transcripts last only a few minutes inside cells. So any damage that's incurred by one error is gone, other than the proteins it produces, of course, after a relatively small amount of time. Now, I'm going to move on to another topic now, and this has to do with the texture of genomes in different organisms, the origins of genome complexity, and how it is driven by non-adaptive mechanisms as opposed by selective mechanisms. The general point here will be that gene structural diversity and genome size passively increases when you decrease the effective size of a population. Again, due to natural selection's reduced ability to eliminate excess DNA. So this is sort of a cartoon view of a typical prokaryotic gene, no introns, very little intergenic DNA, very short regulatory regions, in some cases none between genes. And then you get into eukaryotic genes, very complex, typically half a dozen or more introns subdividing the coding exons, large upstream untranslated regions, very large upstream intergenic DNA, and so on. So much more complex genes, more genes, of course, lots of mobile elements, and so on. And the point to think about here is that every time you add an embellishment to a gene, you're increasing the ways you can break a gene. If you want to add an intron to a gene, now you have to maintain specific sites here that can be recognized by the machine that we call the spliceosome, because these are transcribed along with the rest of the DNA, must be spliced out precisely to maintain an open reading frame. So excess DNA is dangerous. When you look at the scaling of various parts of genomes with genome size, you can see this general gradient that I've talked about here. So as we increase the genome size of organisms here, going from way down here is where bacteria would be to out here where we multicellular eukaryotes live. Down here, this is be 100% of the genome is coding DNA, and we hug that up to a certain point. And then beyond a certain point, the genome size is increasing, but it's not due to an increase in coding DNA. It's due to a sudden increase in intron DNA, pseudogenes, all kinds of transposons, and so on. As we move to the right, the effective population size is going down. Natural selection can't eliminate these kinds of changes from occurring passively in genomes. And I used to think that most of this was due to this mutational penalty, but there's also an energetic penalty of adding excess DNA to a genome. And so we've spent some time trying to figure out how we can estimate the cost of a gene or just a segment of DNA and what it means to the organism. And the way to think about it would be illustrated here. We, of course, have a carbon and energy. We eat food and we have carbon and energy sources. It takes energy, ATPs, the universal currency of energy, to acquire nutrients. And then we build building blocks like amino acids, lipids, and nucleotides, and so on. And these go down these pathways that also consume ATP to build these building blocks. And if you take glucose, for example, and don't squeeze all the energy out of it for operating the cell, instead send the precursors down this path, you've lost an opportunity to get energy from your glucose molecule. So we call that an opportunity cost. Once you build these, put these building blocks together, you want to put them together, that also takes energy to build a polypeptide chain or a chain of nucleotides. Finally, we get to the trait we're interested in, and there's maintenance costs. So what we're very interested in doing now is going through an entire cell, everything the cell does, and trying to figure out the energy budget and how much it costs to do different things in cells. So we have to start with the cost of the building blocks, and we can get that from biochemistry textbooks. For example, the average amino acid, to make it, it costs about 30 ATPs, the average nucleotide about 50. 2 times that 50 gives you that 100 I talked about a few minutes ago for a nucleotide pair. Lipids are very expensive. Peptidoglycan we use in cell walls, for those of us who have cell walls. That's also quite expensive. So the idea that we're trying to get after here is what penalty do we pay by introducing new DNA? Some of it might be transcribed, some of it might even be translated. What is the price to be paid? And does that price for just operating a gene exceed the benefit that we might get out of it ecologically? So the cost of a gene comes at 3 levels. The DNA level, including histones, if you're a eukaryote, RNA, how much it costs to transcribe, and then translation level cost. And what we're interested then is the net advantage of the gene. There's going to be some perhaps selective advantage due to ecology. The gene puts you in a better situation, but we have to subtract from that the cost of just maintaining the gene. So imagine a situation on a fitness scale. There's some fitness level, the gene is absent, and now we add it, but this gene has no advantage. So it has a selective disadvantage, that's the cost. But let's suppose it has ecological benefits. So that's the direct benefit. What matters is the difference between the disadvantage and the advantage, and that's the net benefit. And so we're interested in knowing how big is this quantity here relative to the power of random genetic drift. And all of this has to be divided by the total cost of building a cell, because if your gene costs 100 ATPs, let's say 1,000 ATPs, for example, if the cost of the entire cell is 10 to the 10th, that cost of a gene is a lot lower than if a lot higher than if the cost of building a cell were, say, 10 to the 20th. So we need to know the cost of building a cell. And there are ways to do this by chemostats, and people did this for years in physiological microbial biology. The idea is quite simple. You grow your bug in a chemostat. You control the growth rate by controlling the dilution rate. That is equal to the growth rate. You know the amount of glucose going in, you know the amount coming out, you know the number of cells in here. So you can figure out the rate of consumption of your limiting resource per unit time at different growth rates. You can then plot this as a function of the growth rate. And this has a nice simple meaning. The slope of this is the amount of resources it takes to build a new cell. And the intercept here at 0 growth rate, that's the baseline maintenance cost. That's what it costs to just keep a cell alive, and there's nothing left over to grow. And so we've been able to then take these estimates from a wide variety of organisms. I did this with a former member of my lab, Jordi Marinov. And here's the summary of the cost of growing a cell of different sizes and of maintaining cells of different sizes. They're isometric. The slopes are on a log-log plot are not significantly different from 1. And moreover, the scaling here is continuous between bacteria, unicellular eukaryotes, and multicellular eukaryotes. So now we know we have a general expression for the cost of a cell in terms of energy. That means for a few organisms at least, we can now think about the cost of a gene at all 3 levels, DNA, RNA, and protein, and scale that against the cost of the whole cell and think about how the organism feels this. So not surprisingly, one of these organisms is E. coli. And what we're looking at here are the frequency distributions of the cost for all 4,000 genes in E. coli. So if we just look at the blue line, that's the sum of all the costs. And this is a log scale. So this is telling us that the mode cost for an E. coli protein coding gene is about 10 to the 5th ATPs. So that's about the average cost of the gene in E. coli. Now we divide that by the cost of the entire E. coli cell, and that's what these numbers up here. That's the fraction of each gene's contribution to the total cell's energy budget. And you can see the average is about 10 to the -5. This is important because this line here is the drift barrier. E. coli has an effective population size of 10 to the 9th, 1 billion individuals. And so the reciprocal of that is the drift barrier. Anything to the left of that line can't be felt by natural selection. Everything to the right of here can be. What this is telling us is that for a bacterium like E. coli, even a single base insertion can be felt can be perceived by natural selection. This helps explain why bacteria have such streamlined genomes based on basic bioenergetic principles. 2 other organisms we can look at now that have similar data, yeast and the model plant Arabidopsis, and 3 things happen. The absolute cost of genes go up. We have introns to worry about here. We have histones on the DNA. So genes are much more expensive to make in eukaryotes. These go up. On the other hand, the cells are much larger. So the denominator, the cost of the entire cell, goes down. That means these relative costs up here go down. The effective population size goes up. So the drift barrier moves to the right and everything starts to move to the left. And what this means is that you can insert even 10,000 bases or so in an Arabidopsis genome, and it can't be perceived by natural selection in this plant. And similar arguments can be made for other organisms. So this provides a natural explanation for how genomes can just passively grow in size simply because natural selection can't resist the insertions that occur over evolutionary time. So I got started quite late. I'm not going to finish my complete talk. I'll finish with one more aspect of the scaling with organism size, bringing us back to the second slide. And this is the scaling of maximum growth rate with organism size in different organisms. And what we're going to see is these data are not actually consistent with the biophysical arguments of power law scaling of 1/4 or 1/3. They are consistent with the idea that, again, that larger organisms have smaller effective population sizes and passively accumulate growth rate reducing mutations. So we've gathered a lot of data over the past several years. These are the maximum growth rates as a function, again, on a log scale of the size at maturity. There's about 20 log differences along here. And I think you can see the general pattern. So interestingly, in bacteria, as bacterial cells get larger, the maximum growth rate actually increases. But in eukaryotes, it decreases, and it doesn't matter what phylogenetic group you're looking at, it continues to decrease into larger and larger organisms. So all these are unicellular eukaryotes, and then we get into some multicellular species. The slope here is -0.2 for heterotrophic organisms, and it's only -0.1 for phototrophic organisms. So very different than the kind of scaling relationships people focusing on biophysical laws have told us. As we move to the right, larger organisms, the effective population size is going down. So we think that the reason this is going down is that as you move to the right, growth reducing mutations are accumulating. And so the idea would be if you were able somehow to increase the effective population size, all of this would shift back up and we'd have a flat relationship. But again, this is the drift barrier imposed by a reduction in effective population size. The way to, I think I'll skip over this graph to the upper right, but just try and explain down below what we think is going on. We're looking at situations where there's genes with 3 or nucleotides with 3 effects. So the large ball here means that's a nucleotide site with a very large effect, that it's a medium effect and a very small effect. And a black ball means that you're fixing the beneficial allele in the population. So if we have a very large population, we have almost none of these empty balls. So natural selection is promoting good alleles almost everywhere across the genome. Now we reduce the population size. This major locus is still perceived by natural selection, but now a lot of these other ones start to drop out. Natural selection is not as strong. And then we get down to small population sizes. And the only thing it can hold on to is this nucleotide site with a very, very large beneficial effect. So that's what we think is going on as we move further and further to the right. We're dropping out in terms of beneficial mutations in the population. So because I got started late, I think I'm going to just skip to the save you some molecular biology and math and skip to the final conclusion slide. And the main point I want to make is that what occurs in evolution is dictated by what natural selection cannot do in different phylogenetic lineages. Natural selection is always searching for perfection, but this is limited by the granularity of mutational effects. Most mutational effects are very, very small. It's limited by patterns of mutational bias. It's limited by the power of random genetic drift. The interesting thing is that all levels of biological organization, we expect mean phenotypes to scale with effective population size alone. So that means organisms under identical selection pressures, living under identical ecological circumstances, if they have different population sizes, we can expect them to exhibit phenotypic divergence for purely genetic reasons, not because of natural selection favoring these differences. And what I've shown you is that the reduction in effective population size is caused by increased organism size. It leads to an increase in the mutation rate. It leads to a passive expansion of genome size and gene architectural complexity, the gradient being from very, very streamlined genomes and microbes to very bloated genomes and multicellular species. And it leads to a reduction in maximum growth rate of individuals. So we think this is a very, very general hypothesis that applies to all traits. And if the hypothesis is correct, we need to get further comparative data for a wide variety of traits, but we expect to see these gradients continually show up for traits even down at the biochemical cellular level. So I'm sorry I wasn't able to come to Armenia. This is a picture of my lab in pre-COVID days. We can't stand next to each other like this. You know, we're all wearing masks and standing 10 feet apart, but a lot of people have contributed to this work in my lab over the past 3 or 4 decades, and we've been very fortunate to have good funding from a wide variety of funding agencies. We work in Arizona. This is the Center for Mechanisms of Evolution where I work, and this is what it looks like in the general area around here. We're trying to integrate cell biology, biophysics, biochemistry, and population genetics, and developing a new way of thinking about evolution.